As we've mentioned before, respiration includes four processes: pulmonary ventilation, external respiration, the transport of respiratory gases through the blood, and internal respiration. Here, we want to start to take a look at how these gases are transported through the blood. We'll focus on transporting oxygen here, and we'll visit transporting carbon dioxide in a future discussion. Molecular oxygen is transported through the blood in two ways: dissolved directly in the plasma and bound to hemoglobin within erythrocytes. Oxygen itself does not dissolve well in water, and because of that, only about 1.5% of our total oxygen load is transported dissolved in the plasma, which means 98.5% of our oxygen is transported bound to hemoglobin within red blood cells or erythrocytes. Hemoglobin is made of 4 polypeptide chains. Each of these chains contains an iron-containing heme group. Each iron can bind to one oxygen molecule. This means that each hemoglobin can carry 4 oxygens attached to it. When oxygen is bound to hemoglobin, that hemoglobin is referred to as oxyhemoglobin. When it's unloaded, or oxygen is not bound, hemoglobin is referred to as reduced hemoglobin or deoxyhemoglobin. When one heme group within that hemoglobin binds to oxygen, it causes the entire hemoglobin protein to change shape, which makes it easier for the remaining hemes to also bind to oxygen. The reverse is also true. When one heme group unloads its oxygen, it also causes a three-dimensional conformational change in hemoglobin that makes the other heme groups lose their affinity for oxygen and unload. This makes both loading and unloading with oxygen quick and efficient. The rate of loading and unloading is also affected by partial pressures of oxygen, temperature, blood pH, partial pressures of carbon dioxide, and the concentrations of a chemical called BPG that circulates through the blood. Let's take a look at the influence of the partial pressure of oxygen on oxygen loading and unloading onto a hemoglobin. Heme has a high affinity for oxygen when oxygen levels are high, like in the arteries of the cardiovascular system. In this case, because there is such high oxygen content, the heme groups bind tightly to oxygen and carry it through the body. However, heme has a much lower affinity for oxygen when oxygen levels are low, such as in capillary beds as oxygen starts to get unloaded, and in veins. Because of that, as oxygen levels in the blood start to drop, it actually causes oxygen to get released from the heme groups of hemoglobin, further increasing this unloading process and helping to ensure that oxygen only gets unloaded where it is needed most, in those parts of the body where oxygen levels are already low. Hemoglobin's affinity for oxygen is also reduced by an increase in several other factors, like temperature, carbon dioxide levels, the concentration of a hydrogen ion. This is the ion that actually makes something acidic. Keep in mind that as we increase acidity, it actually causes the pH number to get lower. And an increase in BPG also reduces the affinity for hemoglobin to bind to oxygen. The reverse is also true. Oxygen binds more tightly with decreases in these exact same factors. As the body temperature starts to cool, it causes hemoglobin to bind even more tightly to oxygen. As a decrease in carbon dioxide happens in the body, oxygen binds more tightly to hemoglobin. The same thing with acids. As the body becomes more basic, then hemoglobin binds much more tightly to oxygen. We can take a look at these factors using some graphs. Here, we can see the affinity for hemoglobin to bind to oxygen at several different body temperatures. This curve right here is showing our normal body temperature and the affinity that hemoglobin has for oxygen. If our body temperature is increased, let's say during a fever or during exercise, it actually reduces the affinity of hemoglobin for oxygen and causes oxygen to get unloaded at a faster rate. At reduced body temperatures, we can see that hemoglobin binds much more tightly to oxygen and doesn't unload as quickly. In colder kinds of climate or colder body temperatures, we find oxygen bound to hemoglobin at a much higher rate and a much higher affinity. If we take a look at carbon dioxide levels, we can see carbon dioxide levels also affect the binding of oxygen to hemoglobin. This middle curve is showing our normal partial pressure of carbon dioxide of about 40 millimeters of mercury. However, if we increase carbon dioxide levels to 80 millimeters of mercury, we can see that hemoglobin unloads oxygen much more quickly. That means that hemoglobin has a reduced affinity for oxygen if there is increased carbon dioxide. The reverse is also true. If we decrease the partial pressure of carbon dioxide, let's say we cut it in half to 20 millimeters of mercury, then hemoglobin binds much more tightly to oxygen. It has increased its affinity when we decrease carbon dioxide pressures.